Well, welcome back, ladies and gentlemen. We are going to focus this third video on a very, very important process, that is protein synthesis. So what we're really going to focus on today is how does the DNA that is inside of our cells end up coding for the proteins that are necessary for survival inside of our body. It's a relatively complex process. We're going to simple it down as best we can. There'll be a little bit of, I don't say any complicated math. There's a teeny little bit of math is involved with this particular process, but you'll see how easy this really is, and we'll move on from there, all right? So hopefully by the end of this video, you'll be able to understand the process of protein synthesis. Hopefully you'll also be able to differentiate between transcription and translation, two very similar processes. You should also be able to determine the amino acid sequence when given a sequence of DNA nucleotides. So we're going to be doing a little bit of work here, a little bit of math, but I think you guys will understand it just fine. So let's go ahead and get moving on. So we have quite a few key vocabulary terms for this particular unit. The first one being the central dogma of biology. Dogma meaning like key belief or idea. We have RNA, transcription, translation, RNA polymerase, mRNA, TRNA, splicing, codon, anticodon, intron, and exon. So to start off with, we will start with the central dogma. Now, the central dogma describes the process of making proteins. This is a really important concept in biology because they call this the central dogma because it really is central to all life forms. That all life forms use the DNA that's inside of their cells to make proteins that are necessary for life. Without this process, life would cease to exist. And so the process in which this occurs is that the DNA is transcribed into RNA. So we make RNA from our DNA. And then the RNA leaves the nucleus and then is translated at the ribosome in order to make proteins. So the central dogma basically states that DNA is transformed into RNA, or RNA is copied from DNA, and then that RNA goes to the ribosome and makes proteins as a result of that. So we might as well talk a little bit about RNA since we're here and we're talking about protein synthesis. RNA is very similar to DNA, except there's a few key differences here. RNA contains a ribose sugar instead of a deoxyribose sugar. Instead of thymine, we have something called uracil. So uracil replaces thymine. Instead of A pairing with T, A would pair with U. And also, RNA most commonly is single-stranded, at least in eukaryotic organisms. We're not going to find double-stranded RNA. So just keep in mind some of the key differences here between DNA and RNA. So, once again, there are two steps to the entire process of protein synthesis. The first one is transcription, where we're going to take DNA and we're going to transcribe it into RNA. And the next step is that the RNA is going to go to the ribosome and be translated and converted into protein. So how does the first step occur? Well, it's very similar to DNA replication in that DNA is unzipped using DNA helicase. And RNA polymerase, not DNA polymerase like what we talked about in replication, but RNA polymerase is going to add nucleotides to make strands of mRNA. The M stands for messenger. And that mRNA molecule is going to carry the message from the DNA to the ribosome. Now, all of this occurs inside of the nucleus. The RNA polymerase adds A's, U's, C's, and G's. So, for example, if the DNA, there's a T, it's going to add an A. A still kind of pairs with T. But when we get an A on my DNA, a U is going to be placed on my RNA, as you can see in the picture here. So you can see we've got T, and then so the RNA is going to have A. We have an A in our DNA, so that's going to pair up with U, C pairs with G, G pairs with C, A pairs with U, G pairs with C, and A pairs with U, and so on. So keep that in mind that we will not have any T's in our RNA, but rather we're going to have U's instead. So keep in mind that lots of DNA is kind of junk. It really doesn't code for anything. And so since RNA is a direct copy of DNA, there's going to be a lot of RNA that is junk as well. And so what we do is we perform the process known as splicing. And splicing removes the junk DNA that really isn't important. Exons are what we call the pieces of RNA that are important. So those are the pieces that we keep. The introns are the pieces of RNA that are removed because they don't necessarily code for anything. And RNA ligase actually chemically bonds the exons together to form our final mRNA strand. Now, all of this is done inside of the nucleus, and then that final RNA can actually leave the nucleus and head to the cytoplasm for the next process. So keep in mind that there's a lot of processing of that RNA molecule. It is spliced, meaning we remove the introns in the nucleus. That's how I remember it. Introns stay in the nucleus. And the exons get to exit the nucleus because they're the part of RNA that is important for coding our protein. So something we need to keep in mind is that the sequence of RNA is broken down into groups of 3 called codons. So every 3 letters represents a different codon, which is representative of an amino acid. So if we take a look at this chart here, we have 20 different amino acids, and some of them have just one sequence of 3 letters, some of them have multiple sequences. But each 3 nucleotide sequence codes for a specific amino acid on that chart. So if we look, AUA codes for isoleucine, UUU codes for phenylalanine, and so on. Amino acids are the building blocks for proteins, so each 3 letters of RNA codes for a specific amino acid, and those groups of amino acids, so we get multiple amino acids, they make up a protein, and that's based on the sequence of amino acids that exist. So different proteins will have different sequences of amino acids. So what happens in the next step? Well, the mRNA leaves the nucleus for the ribosome, and then the mRNA attaches at the ribosome and is read through 3 nucleotides at a time. TRNA, which stands for transfer RNA, reads each codon and starts an amino acid chain. So tRNA actually attaches to the mRNA. Now, if you take a look at the codon here is AGU. Now, notice on the tRNA that it is complementary to that. It is UCA. And that is really important because that tRNA is what carries the amino acid to the ribosome in order to start making your protein sequence. And this process continues with each 3 nucleotide sequence. So you see the next triplet code is CGU. Well, the tRNA is going to read GCA, and then another amino acid is going to attach to that protein. And this process continues. ACG, UGC is going to be on that tRNA, and it's going to bring an amino acid. Now, this starts with a start codon. There's a specific codon that starts every single protein sequence, and that is AUG. So that initiates and starts the sequence because it codes for methionine, which is our start amino acid. And this process continues until it reaches what is known as a stop codon. There are 3 stop codons, and then that tells the process to stop. When that tells the ribosome that we are done making our protein, and then the mRNA detaches, and then the ribosome goes away. And then we have our protein when we are done. Also take a look at the protein sequence that is up at the top. We also call this a polypeptide. That's another name for an amino acid sequence. And the chemical bonds that hold these amino acids together are known as peptide bonds. Now, so if we take a look at the code here, we can get a better understanding of how this process works. So if my DNA reads TAC, CGC, CAA, ACC, GAA, TACT, we could break this down into mRNA as AUG, GCG, GUU, UGG, CUU, UGA. I know I just added a lot of letters out at you, but we break these down into groups of 3. So you have AUG. I look at my chart. I look up AUG. That codes for my start codon. Keep in mind that keep in mind that mRNA sequences may have many letters before AUG actually starts the process of making the protein. We keep going, so GCG, I look at my chart, that codes for alanine, GUU for valine, UGG for tryptophan, CUU for leucine, and then we get to UGA, which is our stop codon. The stop codon tells the process to stop. Now, this is just a shortened version of this. Many of the proteins inside of our body may have hundreds of thousands of amino acids in their particular sequence. So it's very important that we make sure that we get this correct and we get this accurate inside of our body. Otherwise, we could encounter some very serious problems for the cell as well as our body. So it is a bit of a complex process, but please do keep in mind that the central dogma is the process in which DNA is converted to mRNA, and then that mRNA is used to make a protein. There are two processes for this. One is transcription, and the second one is translation. And then using the codon chart, we can easily determine the amino acid sequence from a sequence of DNA. So keep this in the back of your mind. We'll spend some time going through this and spend some time practicing with it in class. Hope you have a great day, guys. Hopefully this makes sense to you, and we will talk to you later. Bye-bye.